Alrighty, so welcome everyone to our November Coast to Coral online seminar series. This is going to be the last one for 2023. And thank you for joining us online. We might have a few more coming in and out as the night progresses. The recording is also going to be recorded just so that you know, and it'll go up on our YouTube and social media pages later in the week. So before I begin, I'd just like to acknowledge the traditional owners of the lands and sea country on which we live and work. So I'm currently on Palawa country in Tasmania, and I just want to acknowledge all their elders, past, present, and emerging. So tonight we have Matt, who's joining us to talk about isopods in the marine environment. I met Matt last year on Lizard Island, of all places, so I'm really excited to hear about some of the work he's doing. And I'll give an introduction of Matt later on. But before that, I just wanted to give a brief overview of Reef Check Australia for anyone that might not know. I think everyone that's here tonight is a regular, but for anyone that doesn't know, we're a not-for-profit organization, and we do a lot of hands-on research, which includes monitoring of reefs through citizen science. And on top of that, we do a lot of outreach and educational activities. So for example, this seminar series is a great example of that work. So for tonight, we just ask that everyone keeps their video and mute off while Matt is speaking, just so that we're not interrupting him or anyone else that might be speaking. If you want to find out more about any of our activities or events, or this seminar series in particular, which will be restarting in 2024, feel free to message the email here, secsurveys@reefcheckaustralia.org, or feel free to head on over to any of our socials. The same information is there. I'll probably do a group photo later in the night just in case anyone joins us. So our upcoming events are pretty quiet over the next couple of months, so keep an eye out on socials for any events that might pop up ad hoc in the next 2 months, but really it's pretty quiet as we go into the Christmas and New Year period. There might be some small community events in Southeast Queensland, Townsville and the Whitsunday regions, and we'll probably have ambassador and surveyor training coming up next year. So I know we've had quite a few people emailing us or contacting us via Facebook about those types of opportunities and how they can get involved. So there'll be a lot of information about that coming in the new year. So usually beginning of the year can be quite busy and a lot of opportunities to get involved. And of course, we just want to give a massive thank you to everyone that's involved, from staff to volunteers, and to anyone that's just come along to any of our events or supported us at stalls. We're really appreciative of it, and we just really wouldn't be the organization we are without all of your help and support. So as I mentioned, we won't be having an online seminar series next month or in January either, so we're having a bit of a Christmas break. I will say we are often looking for speakers, and they tend to be more sort of biased towards the Brisbane and kind of Sunshine Coast region, just because that's where some of our team leaders are based. So if you know anyone that might be willing to speak or talk about any of their work, as long as it's kind of in the marine environment or marine research related, so it can be, coastal wetlands to coral reefs to even Antarctic science, we're more than happy to hear about that work. So if you want to dob a friend in or a colleague, let us know. And of course, if you want to get involved as a volunteer or even more casually, or whether it's, you want to get hands-on and help us out with our surveys, please head on over to any of our social channels. So Facebook and Instagram are the ones where we're particularly active, and you can find out more about our events and ways that you might be able to get involved. And of course, I'd just like to thank all of our supporters. So from all of those organizations that support our volunteers or fund any of the work that we do, we're really grateful for that, because it's what makes all of this possible. And of course, the team behind a lot of these events that we hold. So the seminar series does take a lot of work behind the scenes, getting speakers and organizing content. So I'm really appreciative to everyone that's helped in making that possible, particularly this year. And so the moment we've all been waiting for, so we have Matt Nicholson talking to us today, and he's a marine ecologist from Miami, Florida. As I mentioned, I met Matt last year on Lizard, and I'm pretty sure this photo is taken at Lizard, at least it looks like it. And he was doing his PhD back then, but now he's got an awesome postdoc position at the University of Miami, where he's still studying isopods, which is what he'll be talking to us about tonight. So his research really focuses on the interactions between coral reef fishes and marine parasites. Specifically, his work aims to determine how parasites fit into marine food webs, given that they have often been ignored in marine food web studies. While his lab collects data all over the world, most of Matt's work has focused on reefs in the Caribbean and, of course, the Great Barrier Reef. And I just want to say before I hand over to Matt, he is joining us at 3:30 a.m. his time over in the States. So we're really grateful that he's still accepted to do this. We're really, really grateful, and we're very excited to hear what he has to say. So over to you, Matt.
Cool. Let me just share my screen. Share the right screen. Okay, so everybody can see the PowerPoint, yeah? Hopefully?
Yep.
Okay, sweet. All right. So first of all, thank you so much for having me. I'm really happy to stay up late and do things like this. So I was looking forward to this all day, really. And you know, I've got a nice strong cup of coffee, so I'm ready to go. My name's Matt Nicholson. I am from the University of Miami. That's where I'm doing my postdoc right now. That's also where I finished my PhD. It's not where I started my PhD, but I'll tell you a little bit about that in a second. But today I'm going to talk about our curious little critters called Gnathiid isopods, which are ectoparasites on coral reefs. I like to start with this quote from E.O. Wilson that says that there's no better high than discovery, because really this quote sums up why I love doing science and research, and why really I don't think I could do anything else in the world. I think that this is the only profession that I really could sink my teeth into the way I do research, because to me there's just no feeling like collecting data and feeling like I'm discovering new things, you know, all the time. So I call myself a trophic ecoloparasitologist, because I began as sort of a trophic ecologist, right? So that's sort of studying food webs and predator-prey interactions and those sorts of things. But then when I got into my PhD, I started more blending parasitology with ecology. So now I have this this new term that I have come up with for myself, because traditionally parasitology and ecology have been very separate fields, and parasitology is focused more on you know, direct effects on individual organisms, often as it pertains to human health. And for a long time, you know, parasitology as a field didn't really blend with ecology and other fields of science. And I have to thank Brooke Mitchell, who took this picture of me at Lizard last year. And it's like one of the coolest pictures that's ever been taken of me. And I'm going to put it in every talk I ever do for the rest of my life. So a little bit about me and my journey. I like to talk about this for a second, just because I feel really lucky to have been able to do the things that I've done so far in my career. I was born and raised in Miami, so I kind of have also come full circle. But I was born and raised in Miami. I did my undergrad on the west coast of Florida, and it was there where I kind of got my first taste in research, with a field course to the Bahamas. after I finished my undergrad, which actually I was a pre-med when I did undergrad, and it wasn't until like halfway through where I really kind of realized that marine science was really an option and not just a thing I liked as a kid. Once I finished, I took some time off from sort of school, and I went and worked in South Africa. I worked with great white sharks there for a little bit, and then I volunteered in 2012 actually with a parasite lab that I heard about through a mutual friend, and I did one little like 2 week data collection trip to Puerto Rico. And I thought that would be the only experience I ever got with parasites, because I had just gotten done working with great white sharks, and that's all I was ever going to do for the rest of my life, is work with sharks and big charismatic megafauna. And then after that, shortly after that, I did my master's at the University of Exeter in England. So I kind of went over the pond there for a year. I studied seabirds while I did my master's, actually. Then when I came back, I started my PhD working with parasites and parasitic symbioses here, not actually here in Miami. I actually started my PhD in Arkansas, which if you don't know much about Arkansas in the States, I don't blame you. Arkansas is actually a landlocked state, but my advisor used to be a professor there. Fortunately, we left Arkansas and came to Miami. I much prefer Miami. And so I transferred from Arkansas State University to the University of Miami, where I finished my PhD. And now I've started a postdoc here in the same lab. And it was during my PhD where I got to visit Australia for the first time. And since then, I think I've been out to Lizard 4 or 5 times, and I think we're planning on going again next year. Now, parasites, right? So parasitism is the most common animal lifestyle. Roughly 40% of all animal species are parasites. But despite the fact that so many organisms are parasites, they often get ignored in a lot of different ecological studies or biological studies, community level studies. They just kind of get left to the side. And I put up, didn't mean to skip that. I put up these kind of pictures of some nice megafauna. This picture here of the zoomed in marlin was actually supposed to be on a transition. It was a zoomed out picture. You were supposed to see the white shark first. Sorry, I flubbed that up. But this is going to be the only charismatic megafauna that we really talk about today, because today is more about the charismatic microfauna. But I wanted to show everybody a nice turtle, a nice shark, and a nice billfish, because even though they're cool and I appreciate them, to me, they're really just kind of big vessels for parasites. Almost every organism that you can think of has parasites, and even some parasites have parasites. And so our lab here in the Parasite Ecology Lab, we work a lot in the Caribbean. I'll show this map again a little bit later when I talk about one of the specific projects that we're working on right now, but we kind of work all over the Caribbean at several of these different islands. And really, we try to study all aspects of the ecology of various parasitic interactions, and sort of how they fit into the greater community of organisms. We also, of course, work in Australia. All the work that I've done with the lab has been, of course, out at Lizard Island. We do have plans to visit some other areas around Australia. I don't know which ones yet. We haven't quite gotten to that part in the planning. But in addition to the Caribbean and Australia, our lab also does work in the Philippines. We do some work in South Africa. We have kind of collaborators really all over the world that we work with. But most work is in the Caribbean, in Australia, and a bit in the Philippines. So how do you study a small marine parasite? Because really they're out there, right? They're all over the place, but you can't catch them like you would a fish, right? So in order for us to catch them, we mainly use light traps. So I don't know if you can see, you probably can't see my my cursor, but on the left side of the frame here, you can see my hand in the frame of the picture, in that top left picture, and I'm holding a light trap. And actually, I have one here. So we make these light traps out of big PVC bodies, and we put funnels on either end of them. And then we put a flashlight inside the PVC body and close it all up. And then that sits out on the reef overnight and collects all sorts of different plankton. We pick them up the next day, and then we sort through all the different contents and pick out the parasites that are in there. They're just kind of in there mixed in with all the plankton, and just kind of like insects to a light on land, they're attracted to the light and they get stuck in the trap. We also, to catch parasites, and specifically our Gnathiid parasites, we use caged fish. So we'll catch fish on the reef. The bottom right pictures are kind of some poor pictures of me catching. We use all different kinds of nets. We use cast nets and we use barrier nets, which, if you're not familiar with the barrier net, it's a net that is weighted on the bottom and has floats on the top. So it forms kind of like a wall. And you can kind of corral fish into it and then scoop them up with a net. So we use barrier nets. We use all different kinds of nets. And really the kind of net you use is dependent on the kind of fish you're trying to catch. And so what we'll do sometimes is we'll catch a series of fish, and then we'll put them in mesh cages. And then those cages go on the reef for a set amount of time. And then we essentially use the fish as bait. And even in as little as 30 to 40 minutes, our Gnathiid parasites will attach to the fish that we put down on the reef. I suppose I should also mention, we use a couple of, these are the two main methods we use to collect Gnathiids and collect parasites. We do also, we've started to make rubble beds, right? So we take a bunch of dead coral and rocks that's pretty porous and has what we call little hidey holes for little critters. And so we'll kind of pack that tight in some pretty wide mesh and make a little bag out of it, and then let that sit on the reef for a couple weeks, up to a month, and then bring that in and try and kind of flush things out of the porous parts of the dead coral. That has been, we're sort of trialing that as to see if it's a viable option to really collect parasites. It's kind of been hit or miss so far, but light traps and cage fish are really pretty aces for us. But now, to talk about kind of the star of the show today, right? So Gnathiid isopods are the most common ectoparasites of coral reef fish. Now, here's our little example coral reef fish. And we refer to gnathids lovingly as the ticks of the sea, because similar to ticks and mosquitoes on land, gnathids remove a blood meal from host fish. So they live in the benthos, right? They spend their time in the bottom in the substrate. Then they emerge to attach to a fish. They attach from anywhere from minutes to hours. There are a couple species that seem to attach for a set of days. They attach a bit longer, but generally it's minutes to hours. They attach. They remove a blood meal, and then at that point they go back down into the substrate. They digest over a period of maybe like 4 to 7 days, depending on a variety of factors. And then at that point, they molt and they emerge again as the second stage juvenile. They feed once again. They go back down, they digest again. You can see there's a pattern emerging here. And then they come out as a third stage unfed juvenile. They feed one more time. And then they go back down and they molt to the adult stage. So they have 3 juvenile stages that they remove and digest a blood meal. Then once they get that third blood meal, they don't ever feed again. They molt to the adult stage, and they're going to ride that final third blood meal until they die. Once they molt to the adult stage, they reproduce, and that's it. The males develop these really gnarly mandibles in the front of their face. So the picture over here that I showed, this guy that's in the center of the frame, that's a male Gnathiid. And then the one next to him is a third stage juvenile. So in just a few days, the head of that third stage Gnathiid becomes the head of the male. So that they grow a new head in the span of like 4 to 5 days. Now, Gnathids are most active at dusk, dawn, and overnight. They're much less active during the day. There are some species that are more active during the day. Such there's one species actually around lizard, um, uh, Gnathiid oromaculosa, that tends to be a little more active during the day. But in general, they're pretty nocturnal. And because of that, when we're out collecting data, we tend to spend a lot of time in the water at night. So for for a lot of the projects that I've worked on, we get in the water at dusk, we get in the water at midnight, at 1 a.m., 2 a.m., 3 a.m. We do a lot of stuff at dawn. When we get in the water at dawn, that normally means that we're getting in the water just before dawn. So it's something like maybe 4 or 5 in the morning. So we work some really funky hours. But let me tell you, there is really nothing like being in the water for sunrise. Some of you maybe have been in the water for sunrise. It really is just kind of a really magical time to see the sun coming up, but also what's going on in the water. The sunrise and sunset times are really these interesting, transition times where the nocturnal fish are kind of going to bed or waking up depending on the time, and the daytime fish are either going to bed or waking up. And so it's these really interesting changeover times, and it's a really cool time to be in the water. And so during peak times of activity, the Gnathids are searching for fish hosts. You can see the fish in the top right hand corner there. All the little spots on him are Gnathids, and all the little dark spots in the bucket that you can see, for the most part, are fed Gnathids. So they're pretty hard to spot when they're unfed. They're pretty transparent. But then once they have that full engorged blood meal, it's typically pretty dark, that you can see in the bottom picture. Those are all individual fed Gnathids that fed off of that grunt. Actually, this little grunt was at Guana Island in the British Virgin Islands. I remember this fish. We put him down. He sat overnight, and in the morning we collected him, and I think he had like 200 or 250 Gnathids, which is a lot. And here's a little bit of a closer up picture. You can see they have these really characteristic eyes. That's one of the main ways that we spot them when they're in kind of a soup of other plankton. Is the eyes really stick out even when they're unfed. They're pretty. Once you have the search image for them, they're pretty easy to spot by those big eyes. And they attach to fish of all different sizes. So they are. Gnathids are generalists. We call them generalists with preferences because currently we've in the Caribbean, we've identified, I think, 81 different species of fish that they'll feed on. So that's a lot of different fish on the reef that can possibly be exploited by them or infested by them, but they do seem to prefer some species over others. Why exactly, we're not 100% sure. We have some ideas, but we're not really sure yet. And speaking of not being sure yet, there's a lot that we're not sure of with Gnathids because nobody really studies them. There have been 3 groups in the world that have consistently studied Gnathids and sort of Gnathid related activity. And that's our group, the Sickle Lab here in Miami. There's a group in South Africa, Nico Smith's lab, that does taxonomy mostly. And then there was, of course, Lexa Grutter's group at UQ, actually, but she's now retired. So now there are, I guess, 2 groups. Even though Lexa is still pretty active in writing, she's not really, collecting data anymore. So really, there's just 2 groups of us, and we collaborate really closely with each other. But there's not really many of us studying Gnathids out there. So there's a lot that we don't know, which is kind of cool because there's so many different questions that you can ask, but yeah, we're kind of the only ones holding it down. And so here in the frame you see another image in the bottom there of a male. Again, you can see that really big head with the really big mandibles there. And then in the top, you see a female. The female develops the eggs inside her gut, and they'll eventually just become little first stage gnathids and will burst out of the female, and then she'll die shortly after. Now, gnathids can have some pretty severe impacts to hosts. So this is in the top left there. This is another angle of the juvenile settlement stage fish that I showed on the previous slide. A single gnathed can kill a settlement stage fish. For juvenile fish, a single gnathed can slow them to the point that their escape response is not as quick as it was prior to being fed on. When it comes to adult fish, they influence behavior. Okay, so like highly dense areas that are really dense with gnathids can sort of influence the movement and spawning behavior of adult fish, right? So some damselfish will actually interrupt spawning. They will pause spawning behavior to go have parasites removed, which I'll talk specifically about cleaning behavior in a second. But so one of the most important things to a fish, to any organism, right, is reproducing. And these fish will be like, all right, I got to put a pause on this so I can go, get myself cleaned of parasites. That's how kind of important, the kind of influence that gnathids can have on a fish is they're going to interrupt one of the most important occurrences in their life to go have them removed. And I don't have it on here, but gnathids they're also vectors for disease transmission, which we still know very little about. These are kind of some of the newer projects that we're starting to develop. But we do know enough to say that they are vectors of disease transmission. Now, a lot of you probably haven't heard of gnathids prior to this, unless you've talked to me or hung out with me at some point. But you probably have seen them without knowing it. If you've ever been diving or snorkeling and you've seen cleaning activity, right? So cleaning activity is where fish or certain invertebrates will pick parasites off of other fish. Most of what cleaning or cleaner organisms are eating are gnathids. They do sometimes pick off dead skin. They'll pick off other parasites. Sometimes they'll cheat and eat some mucus, which the fish don't really like. But most of what they're eating are gnathids. So if you've ever seen fish at a cleaning station being cleaned by gobies or wrasse, depending on where you're at in the world, you've probably seen gnathids and not realized it. So here in these pictures I have here in the bottom left, you have what's called a dedicated cleaner. So we have different sort of groups of cleaner fish. The dedicated cleaners are fish that they get almost all of their dietary material from cleaning. Whereas we have facultative cleaners on the right side. Facultative cleaners are fish that they're thought to kind of clean only sometimes or only during certain life history stages. Excuse me. So a lot of facultative cleaners, for instance, only clean as juveniles, right? So in this picture that I just put up, there's a little juvenile angelfish that's cleaning a parrotfish. This is in Curacao from earlier this year. And then in the middle, we have the cleaner shrimp. So there's also certain species of shrimp that will pick parasites, pick gnathids and other parasites off of fish. And on top of the shrimp there is actually a Gnathiid Isopod. That's another kind of parasite that you've probably seen if you spend a decent amount of time on the reef. These guys are related to gnathids, but of course they're a bit different. They're much larger. I forgot to mention it, but gnathids size-wise are generally between 1 to 3 millimeters long. So they're not very big. I meant to mention that on an earlier slide. Sorry about that. But yeah, so roughly 1 to 3 millimeters long. So a lot of times when we sort for them, we use like a dissecting microscope or something like that. But once you have the search image and you've spent hours and hours and days and years looking at it, we use a really bright light and we can kind of pick them out that way. But they are pretty small. But cleaning mutualisms they're pretty iconic. They're really kind of cool to watch. And and most of what they're eating are gnathids. Now, in the Caribbean, the main cleaners that we have are cleaner gobies. Now, cleaner gobies associate really tightly with live coral. You can see all the pictures on the left there are gobies resting on different species of coral. And then in the bottom middle, there's a goby cleaning a little grouper, and the goby, of course, was resting on coral. Now, one of the things that we've learned about cleaner gobies is when they don't have coral to rest on, they don't clean as often. And here's a video. I don't know how well you'll be able to see it, but this is in Curacao. Again, another video from Curacao. And I hope you can see it. I'm sorry if you can't, but these are some chromis, and one of the chromis in there is getting cleaned by a goby. Interesting though, this goby was not on coral. This goby was just on kind of dead rubble rock reef. But he was still a really active cleaner. And so while one of them is getting cleaned, one of these chromis is getting cleaned, the other two were kind of waiting their turn. And we see a lot of that early in the morning. So when we're out there at dawn, as the sun is just coming up, sometimes we'll literally see fish lined up, almost single file lined up, waiting to get cleaned. So yeah, in the Caribbean, the primary cleaners we have are these cleaner gobies. Of course, in the Pacific, in the Great Barrier Reef, the primary cleaner, the main dedicated cleaner is Labroides dimidiatus here, the blue streak cleaner wrasse. These are some pictures I took. They're not the best pictures. I apologize for that. But these are some pictures I took out at Lizard last year. Almost this time last year, actually, I think it was. Maybe a couple months. It was like maybe August last year, I think it was. But so there's been a lot of work done studying sort of the impacts of cleaners, specifically a lot of work studying the impacts of Labroides here on host or client fish. And so here, I love this video. This was a dive I did again last year at Lizard. This is a big, not the biggest sweet lips I've ever seen, but a decent sized little sweet lips getting cleaned by two adult cleaners. And then the other video here is an eel getting cleaned by a juvenile. So of course the juveniles are really just kind of black and blue, and the adults have more of a white belly, and they have some yellow gold to them. But they're super active cleaners. They can eat thousands and thousands of gnathids a day. Yeah, this was the video on the right was a really cool dive. There was lots of live coral, lots of big fish. It was really, really cool. So cleaners sort of, help to mitigate some of the negative impacts that gnathids can have on individual fish, and they also help on a community level. So reefs with cleaners have a higher abundance of fish. They also have a more diverse assemblage of fish. So they have a stronger biodiversity of fishes in addition to just having more fish around. And also, there was a long-term experiment that was going on and is still sort of going on, where there were a set of patch reefs that half of them were experimentally manipulated to have no cleaners. So all the cleaners from half of the reefs have been removed for 20 years. And the other ones, the cleaners were allowed to stay. And on the reefs with no cleaners, the resident fish had a lower body condition. They weren't quite as healthy as the reefs that had cleaners. So cleaners are definitely, they can help mitigate some of those negative impacts of ectoparasites, specifically of gnathids. Just interacting with cleaner fish lowers stress in client fish also. So having access to cleaners is a huge benefit to reef fish, to fish infested with parasites. Now, of course, there's a lot going on in our oceans, specifically as it pertains to coral. So in a reef where we have a lot of live coral, we typically have a lot of cleaners, we have a lot of fish. One thing I haven't told you yet is, in addition to cleaners consuming gnathids, live coral also eats gnathids. So live coral polyps can consume gnathids. And so when you have live coral, and this example is more tailored towards the Caribbean, but it also holds for Australia. But because in the Caribbean, because of how tightly linked the gobies are to the coral, and specifically their cleaning activity is linked to having live coral. When you have live coral, you have direct consumption of Gnathiid isopods, and then you're also facilitating the habitat for, you also have the habitat that facilitates the cleaning activity by cleaner gobies. So of course, what are we seeing a lot? We're seeing a lot of, well, let me get to that. We're seeing a lot of coral die-offs. Here, I'm pretty sure it made kind of worldwide news. We had some intense bleaching here in Miami, and corals were described, it was heartbreaking. Corals were described to me as melting. It was so warm here. It was like 100 degrees Fahrenheit in some of the waters. And we had massive coral die-offs in some of the most resilient coral communities that we've had here in the Miami area, going on down into the Florida Keys, have just died off in a huge way. And that is sort of a double-edged sword for sort of the state of the kind of, the equilibrium of the parasite community. Is you have less suitable habitat for cleaner gobies to do their thing in removing parasites. You also have potential, less direct consumption by the coral polyps, but also gnathids love dead coral. They love to kind of go inside of the pores of dead coral to molt and to mate. So when you have more and more dead coral, you have more suitable habitat for gnathids, you have potentially less cleaning activity, and you have less direct consumption of gnathids. And what that can result in is sort of a hyperabundance of parasites, to where now the burden on each individual fish is much higher than it used to be. And so if you think of, us when it comes to something like mosquitoes, if we have a few mosquitoes on us, we're okay. We're just fine. But if we have a few hundred mosquitoes on us, we're not going to be very happy. We may have some reactions. We're not going to be in a very good way if we have a hyperabundance of something like mosquitoes on us. And it kind of works the same for fish. They can handle a few. They can handle more than a few, to be honest. But getting to the point where there's a huge density of gnathids is going to have some serious consequences physiologically for reef fish. And I had to throw in my my Anchorman GIF. So here, let's kind of take these two different reefs. On the left is a reef with a ton of live coral, and on the right is a reef with almost no live coral. You have Lizard Island on the left, one of my favorite reefs that I got to dive. I can't remember the name of it. And then in Barbados, the reef I was working on this past year on the right. And you can just imagine that there's likely a lot more direct gnathed consumption on the left, where there's all this live coral, lots of live coral polyps. And on the right, we got so many gnathids in Barbados. There was a really healthy parasite community there. And there's just tons of really good habitat for them there too. So now I've kind of told you a bit about gnathids, what they're all about, what they do. I want to talk to you now about some of the current projects that we have going on. The first part is going to be some of the stuff that I did for my PhD dissertation. Then I'll talk about another couple projects that we've been working on that are kind of cool and exciting and have some interesting results. Some of this I was literally putting together today as we were getting new results in. So some of the stuff that I'm going to share with you here is like brand new, just kind of coming out of the woodwork. So again, I mentioned that I kind of study food webs and food web interactions, and then I got into studying parasites that way. And so I wanted to know, how do parasites, how do gnathids fit into food webs? How do they contribute to food web structure? What kind of role do they play? And so if we, little primer on food webs, if we think of a food web, it's really just a pretty intricate diagram of feeding interactions. Okay. And if you simplify it even further, a food web is really just a bunch of food chains overlaid on top of one another. And so for years now, there's been a lot of researchers advocating for the inclusion of parasites and other underrepresented functional groups, but in this case, specifically parasites, a lot of people advocating for the inclusion of parasites in things like food webs and other community level studies. And there have been some really interesting studies showing that, in fact, parasites play a major role in kind of biomass transfer, in food web structure, and things like that. And one of my favorite ways to sort of show this comes from this paper from 2009. So this is a food web that was constructed from a sub-Antarctic lake. And this one that I'm showing right now is the food web. And so if you imagine each box is a species or a kind of a broad grouping of species. I can't remember what they did here, but essentially, each box is a different group. And then all of the lines, of course, are interactions between the groups. So this version of it here is with no parasites. These are all the interactions between everything that's not a parasite. And this one is when you add parasites in. So there was something like 54% of the feeding links in this system were from a parasite. And so you look at just this one and you think, okay, we have a good idea of what's going on in the system. We can start to figure out, what are kind of the most important predators and what are the most important prey items in this food web here. But then you kind of change the whole picture here when you add in parasites. Yeah, so I forgot I had this in here. So 54% of all the links in the food web ended up involving a parasite. And so now, again, for my PhD dissertation, a lot of what I was looking at was I was looking at parasites, specifically gnathids, as both prey items and as predators, right? So I mentioned that gnathids are, they feed for a very short amount of time. So that means that most of their time they're spending not associated with the host. So they're free living and they're available for consumption, just like any other planktonic species, any other zooplankton. So for the purpose of what I'll talk about today, I'm going to focus on parasites as prey, not so much parasites as predators. That's another study that is not quite finished yet, but hopefully will be finished early next year. But when I was looking at parasites as prey, I wanted to look at what fish out there, in addition to cleaners. So we know cleaners are eating a lot of parasites, okay? We know dedicated cleaners especially are eating a lot of parasites, but what about everybody else? There are so many other groups of fish, so many other planktivores, right? Scrapers, benthic invertivores, detritivores. Are any of them eating gnathids? And so that was one of the big chapters of my dissertation. Was I caught hundreds and hundreds of fish. I think the final count for the Caribbean was 700 something fish. And I essentially looked through their poop. This is the part that my parents were most proud of. I looked through their fecal matter to look to see who was eating gnathids. And you can see in the pictures on the right, these are gnathids that I found in the poop of various fish. And, if you haven't seen many gnathids, you may not think that is obviously a gnathid, But as someone who's seen probably tens of thousands of gnathids, that is very obviously, excuse me, a gnathed. So I spent a lot of my PhD looking through fish poop to try and find gnathids. And what we ended up finding, and this paper is currently in review, was that all of our dedicated cleaner gobies consume gnathids, which was super unsurprising. Our facultative cleaners, which in this case was a wrasse, 64% of them consume gnathids. And then outside of that, it was pretty low. It was interesting that pretty much every group that we sampled, every functional group, did consume gnathids to some degree, but really only the cleaners are eating them in any kind of significant way. And so when I kind of plotted it out, you can see that the cleaners really rise above everybody else in terms of gnathed consumption. So it's interesting that we found that a lot of different groups of fish will eat gnathids and are eating them, even herbivores, right? So it seems like maybe herbivores are incidentally picking them up when they're scraping algae or something like that, because of course that's where the gnathids are resting, that's where they're living. Yeah, there's really nothing that compares to cleaners in terms of gnathed consumption. The closest thing was the planktivores, which makes sense because when the gnathids aren't attached, if they're out swimming, maybe looking for a host to attach to, they're probably going to get picked off because they're going to be swimming amongst the plankton. Yeah, so this is my gift to say that, man, cleaners really can't get enough of those gnathids. And so another project that is this paper is about to come out is looking at how gnathids contribute to habitat connectivity, right? So if we think of there are many diel migratory fishes, right? So these are fishes that they rest in one habitat and then they leave to go forage in another habitat. So in the Caribbean, we have a lot of our grunt species. Around Australia, around lizard, it's a lot of the cardinal fish, the apagonids. So these fish, they rest on the reef in large aggregations during the day, and then at dusk, they leave the reef and they go feed in kind of the sand and seagrass areas for the most part. And so that's, these fish are sort of facilitating a connection between the reef and the seagrass and the sand habitats. But what about the gnathids and the parasites that are attached to them when they leave? So we did an experiment where we put cage fish on the reef and then we took them off when the other fish were leaving for the night. And we found that on average, there's somewhere between 2 to 5, upwards of 40 gnathids attached to these fish as they're leaving for the night. So that means that all these parasites are getting transported from the reef to either the sand or the seagrass to adjacent habitats where really they're not normally very prevalent, but they're getting sort of trafficked there by these sort of migratory fishes. And so it is that most of these fish are only taking maybe a handful, but if you have a reef that looks like this is a reef that I was at in the Bahamas. There are thousands of grunts here that all leave every night to go forage in the sand. How many gnathids do you think they're taking with them, right? And that's every single night. And the final thing I wanted to talk about, the final project that we're working on, this is a big NSF grant that our lab got, that my advisor got, is to just describe gnathed species, right? So right now there are, I believe, 185 species of gnathids that have been described. And we think there are tons and tons more, because again, there aren't many groups out there sampling them. So our current grant is essentially just to go to different places across the world and sample as many gnathids as we can. So previous to this, we've sampled a bit in the Bahamas, and we've sampled a lot in the British Virgin Islands and Puerto Rico. This year alone, we've done, of course, Miami, because it's local. We've done Jamaica, Dominican Republic, Curacao, and Barbados. That's all this year. Next year, we're adding in Mexico, the Cayman Islands, Panama, Trinidad and Tobago, Turks and Caicos, and maybe Grenada. And that's just the Caribbean. We also have some collaborators collecting in Hawaii. We have, I believe, some collaborators in China that are going to be collecting. We should also be going back to Australia next year to do more work around lizard and other reefs in other areas around Australia. But from this so far, just in the Caribbean, we already have 3 undescribed species. So in total, in the islands that we've been to this year, the 5 or 6 islands, we found 6 species. 3 of them were previously described. 3 of them are brand new. And one of them was actually has just formally been described, and we named it after Jimmy Buffett, who of course recently passed away. It was really sad to see Jimmy Buffett pass away, but it was really cool because he heard about the species being named after him, Gnathiid Jimmy Buffetti, and he reacted to it on social media and something like 2 weeks before he passed away. So it was really cool that he got to see that that our group kind of wanted to honor him in this way. And by the way, we also have another Gnathiid species that was described about a decade ago named after Bob Marley. So my advisor is a big fan of naming species after musicians. But these 3 pictures on the right are actually I think the 3 undescribed. There are 3 different Gnathiid species. I think these might be the 3 undescribed ones. But to give you a quick idea, right in between the mandibles, you can see there are these little processes, these little kind of spikes in between the mandibles. That's one of the main identifying features of different species. So in order to morphologically tell different species apart, we have to get males. And so these are actually pictures of 3 different species, and you can primarily tell that again because of that space in between the mandibles and what those sort of frontal processes look like. And so to kind of wrap up, I of course have to shout out all the different people that we work with. We're so fortunate to have a wide network of people all around the world that collaborate with us, in Australia, like I said, the Philippines, South Africa, Hawaii, elsewhere in the States. We're super fortunate to have a really incredible group of collaborators and volunteers that work with us. And I always like to end with this picture. This is a picture of one of my best friends. We were doing field work in Puerto Rico, and we had some downtime, and he wanted to pretend to be a dolphin. And so this is always my reminder that, as a scientist, I'm really lucky to do what I do. And at the end of the day, it's fun, and I really enjoy it. So this is, if I'm ever not having fun, I look at this picture and I remember that I should be having fun. And if anybody has any questions, I'm sorry if I went a little long, but I'd love to take any questions if anybody has anything. And thank you.
Thanks, Matt. That was awesome. Does anyone have any questions? Feel free to pop them in the chat. Since we're a small group, you can also turn your mics on as well.
I have 2 questions.
Yeah, bring it.
A few years ago, myself and another volunteer, Phil, went and did some stuff for Lexa on Lizard Island.
Okay.
So, yeah. And so I remember you said something about how she's finished up most of her field work now, but I was just wondering if you knew if all of her experiments that were set up there, all of like the cultures and the patch reefs are still going in some capacity.
Yeah, so the Gnathiid culture that she had going on there for ages has now been unfortunately shut down. It was a little bit too much to maintain, but the patch reef removals are still happening. So like every time that our group goes, so when like I was there last year, we did removals, and it's kind of yeah, there's several groups that are helping out with it, but it's definitely they're still happening. And
yeah,
when we go back, when we go back next year, we're gonna keep that going, and yeah, I definitely will do whatever I can to keep that project going.
Many hours
at this point it's been going for like
those
20
cultures, picking out gnathids and stuff. And then my other question was, I'm curious to know a little bit more about. Oh, sorry, I think I just cut you off. I'm curious to know a little more about your trip to Australia and where you're thinking of going. I hear there's a really great research station on Orpheus Island, which is one of the Palm Islands, which I won't go into why I know that island, but yeah, just curious what you're looking for, like what sort of habitats.
So, Gnathids really like degraded reef. So anywhere where there are fish and not a lot of coral, we expect to find tons of them. They can be in really shallow reef. They can also be in really deep reef. We normally, focus more on shallow reef just because it's easier. But anywhere where there's, rubbly rock reef and fish, there are gnathids. And so, honestly, if you have any suggestions for places, I'm open to suggestions for places to go, because I know we plan on going back to Lizard. I know there's been talk of going to Ningaloo, but outside of that, like you can see in the Caribbean, all the proximity of the different islands that we go, and we expect to find almost a new species almost every island. So it's also plausible that, even if there's something close to Lizard that I haven't been to, there may be different species there. There's, I think, somewhere around at least 8 species of Gnathiid just around Lizard. Not all of them are actually described yet. So
yeah, that's really
any
cool.
suggestions, please? Yeah,
I might
any suggestions?
flick you an email and take this offline, but yeah, I think
Please.
it might be worth looking at Orpheus, which is run by James Cook University.
Okay, yeah.
Might have a collaborator or have had a collaborator there. That's worth looking into.
Yeah, definitely. My email's on here. Please shoot me an email. Yeah, definitely. That'd be awesome.
There's also really quickly, if there's any research students up this way or that you would be bringing with you, there's also these small $3,000 grants for that region. Anyway, I'll send you an email. We can chat later.
Yeah, that's great. Thank you so much. That'd be super, super cool.
I've got a question.
Yes.
So you said all the fish, like they leave the reef and they take their little gnathids with them to go off into the seagrass. What happens to those gnathids? Do they then burrow into the substrate there, or do they just die? Or
it's a really great question. So that study is has just been accepted, right? So we haven't quite gotten to the next level yet, but that is kind of the natural next question, right? Is we have these gnathids that we know that they're well suited to live in the reef, but then what happens to the gnathiid that gets deposited in the seagrass? Do they burrow in the sand? Do they maybe kind of hang on to the seagrass? Do they just get eaten? We honestly really don't know yet. What we do know, though, is I didn't go into, for the sake of time, I didn't go into too much detail of all the different sort of components of that study. But one of the things we did is we also put caged fish in the seagrass, and then in the morning when fish are returning to the reef, we took them out. And so we did get gnathids attaching to fish that we put in the seagrass. So we know that to some degree they're there. They're in much lower density, but they do seem to be able to sustain themselves somehow. I don't know exactly where they go. Maybe they burrow in the sand. Maybe they, like I said, find something to cling on to. It seems like to some degree they are able to figure something out. But yeah, we really don't know. It's a great question though, and it's something that we probably want to explore in a upcoming project. So
I think Beverly's got a question.
Hi there. My son Kelly, he's got a question for you, Matt.
All right, bring it.
Do the gnathids have parasites?
So Gnathids have, they actually have parasites. They get blood parasites. So it happens sometimes that when a gnathed feeds on a fish and that fish has blood parasites, then the gnathed gets the blood parasites inside of it. And so we don't know too much about any other parasites that gnathids might have. That's kind of some of the only ones that we know, is that they get parasites from the blood of the fish. And then that's how they can transmit disease to other fish, right? So if a gnathed feeds on a fish and gets a blood parasite and then goes and feeds on a different fish, it can give that parasite to the other fish. We also know that gnathids, in this, this has been seen one time, a gnathed fed on another gnathed. So there was a fed gnathed with a big blood meal, and an unfed gnathed fed on the blood meal of another gnathed. So that's called hyperparasitism. So technically, gnathids can be parasites of themselves.
Thank you.
Yeah, no, my pleasure.
Does anyone else have any questions?
I have another question. Are there many instances of them attaching to species higher up in the water column? Or is it mostly like the bottom feeders?
Sorry, I didn't mean to cut you off. That's another really good question. And that's actually a study that that I did as part of my PhD. I didn't mention it here, but I actually, we did a study in Puerto Rico and a couple other islands in the Caribbean, where we actually put a vertical line from the substrate to the surface, and I put clips every half meter, and then I did sets where I clipped a cage fish at different heights above the substrate to see how high above the reef a gnathids would go to attach to them. And we got gnathids, I think, as high as like 3 meters above the substrate, I think it was. Nothing above that, but only, it was like a lot on the ground, a lot at 1 meter, and then it dropped off pretty steeply after like a meter or meter and a half. So most of the action seems to be like, within that first meter to 2 meters above the substrate.
What species of fish were you using for that?
We used, in the Caribbean, we mainly use French grunts. So in the Caribbean, French grunts get slammed more than any other fish we see. Like grunts and snappers get absolutely infested to a degree that other fish just do not. And it's also curious that those are the fish, those are the main fish that leave the reef at night. So the most susceptible fish leave as soon as gnathids are getting most active, and then they come back right as activity dies down. So we go ahead.
What sort of substrate was that off? Was that of like a rubble bed or just like coral reef? Or
it was formerly known as coral reef. It was like a lot of dead little bits of coral, a lot of dead rocky substrate. That's what we have a lot of in the Caribbean. Unfortunately, we don't have much coral left here, but cool. It was that kind of reef. Thanks. But yeah, so most of the action is pretty close to the bottom. It seems that they will venture farther up into the water column occasionally, but not super often.
Anyone else?
If anybody comes up with anything else or wants to chat about anything, please shoot me an email. I'm happy to chat after the fact if anybody's curious about anything, or I'm on Twitter and Instagram and all the socials, and we also have a lab social too, where we like to share stuff. And so yeah, I'm always happy to chat.
Awesome. Well, thanks for joining us so early in the morning. Hopefully you get some sleep after this, or maybe you're going.
Oh no, I'm gonna go get a nice sleep. It's all good. Honestly, I had a blast. I was super happy to do it. I don't even feel tired, if I'm being honest. So.
You look fine. Awesome. Well, we'll share all of this as well on socials, so anyone that maybe didn't make it might get in touch with you if they're interested or want to reach out for whatever reason. But
Great.
yeah, on
Yeah,
behalf of
that's awesome.
ReefCheck, thanks for joining us. If anyone has any follow-up questions, as Matt mentioned, you can give him slide him an email or a message or whatever it might be. But that's all from us. Was there anything else, Julie?
No, I'd just like to thank Matt for a really amazing talk, and yeah, and staying up at 3:30 in the morning.
Awesome. Well, thanks everyone.
It's 4:30 now.
Oh, yay.
Awesome.
Thank you very much for having me. Honestly, thank you so much.
That's all good. Thanks everyone.
Thank you.
Cheers.